Hello, everyone, and thank you for attending today's webinar, Make, Characterize, and Optimize LNPs with Nunchuck Stunner, presented by Unchained Labs. I'm Rebecca Kushmeyer. I'll be moderating this webinar. I'd like to start off by introducing today's speakers. We'll begin with an introduction from Martha Perez, PhD, the Director of Marketing at LNP Solutions at Unchained Labs, followed by a presentation from Julie M. Rumble, PhD, Director of Immunology Contract Services at Cayman Chemical. You can read their full bios on the left side of your window by selecting the Speakers tab. Now, just a few notes before we begin. To access additional resources for today's presentation, you can click the Handouts tab button on the left side of your screen. The webinar is being recorded, and it will be available to watch on demand within 24 hours. We'd love to hear from you during the presentation. Please submit any questions you have using the Questions and Answers tab on the left side of your screen. Time permitting, we'll conclude with a live Q&A session. Now, let's begin. Julie, please go ahead.
About Unchained Labs solutions for LNPs. Lipid nanoparticles are being used in gene therapy, cancer treatment, vaccine development, and many other biopharma applications. They are a versatile delivery vector because they protect their payload and only release it once it has gotten to where it needs to go. This confers multiple benefits, including enhanced solubility, increased efficacy, improved pharmacokinetics, decreased toxicity, and ease of changing out the payload. There are three basic characteristics of LNPs that make them versatile and useful. One is the payload, which can be a variety of different molecules like RNA, DNA, protein, or practically any other active pharmaceutical ingredient. Another is the particle size, which is an important factor in determining the cell or organ that the payload will get to. And today you will hear from our partner, Cayman Chemical, about how different lipids can affect important characteristics of LNPs. Screening for different conditions for LNPs is made easy on Nunchuck. Nunchuck is Unchained Labs' versatile LNP maker. Nunchuck makes screening conditions easy with our total flow rate screening application and a wide volume range of 0.25 milliliters all the way up to 1 liter. That means that it can be used from early R&D screening to a mid-range scale up. So how do we do this? Well, the Nun gets it done. Here is a closer look at the inside of the Nun. You simply place an organic solution containing lipids and an aqueous solution containing your payload into luer-lock syringes. Then you lock the syringes into the Nun. Your solutions are injected into the microfluidic mixing channels of the Nun cartridge. Our patented design causes the two fluids to mix in a controlled and reproducible way, resulting in homogeneous and loaded LNPs. You can use a wide range of both payloads and lipid chemistries to get great results every time. Once you have created the LNPs on Nunchuck, you will want to take advantage of our hassle-free LNP quality analysis that Stunner provides. With low volume and high throughput dynamic light scattering, DLS, to get the size and polydispersity index at the same time. Stunner can quantify your total payload concentration, whether it's RNA, DNA, protein, or another API, without the need for any reagents, standards, fluorescent dyes, or particle disruption. All the sizing and polydispersity data you will see today was gathered on Stunner. But that's not all. Unchained Labs provides killer LNP solutions. Nunchuck makes LNPs with the ultimate control to get quick, reproducible, size-tunable assembly of nano-sized particles to increase process control and decrease both amounts of costly reagents and optimization time. Stunner lets you quickly answer the questions about size and quality of nanoparticles. You can do buffer exchange and concentration on Unagi, and you can test the thermal stability of LNPs with UNCLE. From start to finish, Unchained Labs is here to provide the right tools for the job. And Julie is going to tell us more about how she used Unchained Labs tools.
Thank you so much for the introduction, Martha. As Martha mentioned, LNPs encapsulating nucleic acids are usually composed of 4 lipid components: an ionizable or cationic lipid, a pegylated lipid, a helper phospholipid, and cholesterol. We wanted a way to evaluate uptake of particles we made without using expensive mRNA. So we decided to include a 5th lipid in some preps, BODIPY-labeled cholesterol, as a minority of total cholesterol. Once the lipids are mixed with the aqueous phase that includes the cargo, lipid nanoparticles encapsulating cargo are made. The whole particle will then fluoresce in the green spectrum with 480 nanometer excitation, and cells taking up the particles fluoresce immediately as well. For this project, the workflow for formulation and testing of the LNPs consisted of prep of the ethanolic lipid mix and the aqueous cargo phase. These two were mixed using the Nunchuck with the single-use Nun. Resulting formulations could then be analyzed for particle characteristics such as diameter and polydispersity index, or PDI, which is a measure of the uniformity of the particles, using the Stunner, as Martha mentioned. LNPs were finally added to cells for imaging of uptake of BODIPY-cholesterol. The whole project was divided into 2 phases. For the first phase, shown on top, initial screening of variable flow rates in the Nunchuck using the screening function was accomplished with poly-A RNA and a fixed molar ratio of lipids. These preps were screened using dynamic light scattering on the Stunner for particle characteristics, and selected preps were further evaluated using BODIPY uptake in cells. Once candidate formulations were established using these tools, we moved on to phase 2, shown on the bottom. Bulk formulations were also made on the Nunchuck and used mCherry-mRNA. These utilized the optimal conditions from the screening set and were again made with and without BODIPY. In addition to Stunner particle characteristics, we measured cargo encapsulation efficiency and tested multiple cell lines for uptake and expression of mCherry. We wanted to take advantage of Cayman's library of published ionizable lipids to test how some of these newer lipids would behave in LNP formulations. SM102 and ALC0315 on the left have proven themselves effective already in the COVID vaccines, but different organ targeting is desirable for many applications. So understanding the properties of LNPs generated using different ionizable lipids will be very important moving forward. For example, 113012B in the top right has been reported to target lymph nodes in mouse studies. CKK-E12 and C12-200, both on the bottom, have been reported to induce strong cargo expression in innate cells in the spleen. So starting with SM102 and ALC0315, we made screening preps of LNPs in our Nunchuck. The flow rate ratio, the ratio of aqueous to lipid, has been well established for these two. So we maintained flow rate ratios at 3:1 for these two sets. What I am showing here is the particle characteristic data we collected using our Stunner. We have overlaid 2 graphs of size, shown in bars, in the bars and on the left axis, and polydispersity index, points, and on the right axis. Polydispersity index, or PDI, you'll remember, is a measure of the size distribution or uniformity of the particles. The different colors represent different total flow rates, or TFRs. Nearly all of the preps with both ionizable lipids here, with and without BODIPY, showed size at or below 100 nanometers, and PDIs well below 0.2, which are generally considered to be the cutoff for quality uniform particles. So total flow rate did not dramatically affect particle characteristics for these particular particles. In addition, BODIPY did not overall affect the size or PDI. With the newer ionizable lipids, the particles came together a little bit differently. For instance, CKK-E12 made particles that were still around 100 nanometers, but had slightly higher average PDIs, meaning that they were a little bit less uniform. BODIPY inclusion still didn't change the particle characteristics measured by Stunner, but a change in the flow rate ratio to 5:1 aqueous to lipid, which is in the set furthest to the right, resulted in larger particles. Notably, the lower total flow rates, the orange and black bars, which represent 10 and 5 milliliters per minute respectively, generally made larger particles as well. These data help to determine what the optimal conditions are for LNP formulation with these lipids. Similar observations were made with C12-200 with flow rate ratio of 5:1 and lower total flow rates, on average resulting in larger and less uniform particles. Additionally, 113O12B showed the same trend, with even greater differences with total flow rate and flow rate ratio. You can see that this ionizable lipid made substantially larger particles on average than the others tested in this screening set, but the PDIs were still found to be clustered around 0.2. So, in a relatively short period of time, using Nunchuck and Stunner, we were able to generate and perform initial analysis on nearly 80 LNP formulations, with varying flow rates, flow rate ratios, and multiple ionizable lipids. Of course, while DLS on the Stunner can tell you a lot about particle characteristics, uptake by cells for cargo expression is really the ultimate goal. The advantage to using BODIPY-cholesterol as a component of the particle is the readout of particle uptake in the absence of expensive nucleic acids. This makes for a substantially cheaper screening tool for initial assessment. Here, selected screening formulations that included BODIPY were dialyzed and applied to HUH7 hepatocytes. All of the formulations were taken up, although there were distinct differences between the ionizable lipids. SM102 and ALC0315 were taken up to the highest degree, as measured by imaging analysis, while the higher flow rate ratios that made less optimal particles by Stunner analysis similarly were less than optimal in terms of uptake. Once we had screened our test formulations for particle characteristics and uptake, it was time to make formulations to test expression using the parameters optimized in the screening run. Here, we used mCherry mRNA so we could fluorescently visualize both BODIPY and the expressed protein. We fixed the N to P ratio, which is the calculation of the number of nitrogens in the ionizable lipid and the number of phosphates in the nucleic acid backbone, at 8:1 for these experiments. Since some of these ionizable lipids have substantially more amines than others, the absolute RNA concentration in the aqueous phases varied accordingly. We used Nunchuck's bulk formulation mode and again tested particle characteristics with Stunner, but also tested cargo encapsulation efficiency and evaluated both uptake and expression in vitro. Initial assessment of particle characteristics using Stunner indicated that all of the particles produced were under 125 nanometers, again in the bars and on the left axis. PDIs in the points and on the right axis were all under 0.2, indicative of monodisperse LNP populations. These were better than those produced in the screening runs, although these measurements were made after dialysis. CKK-E12 and C12-200 based particles were noticeably larger than the others. And again, BODIPY, which is shown in red, did not have a measurable effect on the particles themselves. Using Thermo's RiboGreen kit with and without Triton X100, we tested encapsulation efficiency of the mRNA. As expected, SM102 and ALC0315 particles encapsulated very nearly 100% of the mRNA. And additionally, 113O12B based particles encapsulated about 85%. All of these numbers are suggestive of high quality particles. The two ionizable lipids with higher numbers of amines, however, 4 for CKKE12, and 5 for C12-200, we're only able to encapsulate about 50% of the mRNA. This suggests that further optimization of the N-to-P ratio may be necessary. Finally, we assessed the transfection efficiency of each of the LNP formulations on 2 cell lines in vitro. On the top are HUH7 cells, a hepatocyte line. We assessed mCherry expression over time using our Citation 5 imaging plate reader and BioSpa automated incubator. We imaged these every 4 hours over 2 days. In hepatocytes, SM102, shown in blue, clearly induced the greatest expression, with CKKE12, shown in purple, and C12-200, shown in orange, surprisingly also inducing strong expression of mCherry in the hepatocytes. On the bottom, you can see that the results were different in A549 cells, which is the lung epithelial line. The same 3 particles induced expression, but CKKE12 and C12-200 induced more widespread expression than SM102. Actually visualizing just the SM102 induced expression in the 2 different cell lines, you can see that the mCherry fluorescence in the HUH7s on the left appears substantially more intense than that of the A549 cells on the right, suggesting not only more widespread transfection, but higher protein production in these cells. It is also notable that neither ALC0315, which is shown in red, nor 113O12b, shown in green, particles, both of which showed high encapsulation efficiency and other favorable particle characteristics, expressed measurable mCherry in either cell line. Especially since ALC0315 has been proven effective in the Pfizer-BioNTech COVID vaccine, the obvious question is why? It has been suggested in the literature that in vitro results don't always match up with in vivo ones, but also that mouse models don't necessarily reflect human data. These results emphasize that model systems need to be very carefully chosen, and data need to be cautiously considered with regard to LNP-based expression systems. That being said, the data from the newer ionizable lipid CKKE12 are striking, as shown in these comparative images of lung cells on the right and liver cells on the left. The mCherry expression is dramatically more intense in the lung cells than the liver cells. These data support the idea that this lipid holds promise with respect to targeting tissues other than the liver for gene expression with LNPs. Since we included BODIPY in these particles, we could also start to examine the dynamics of uptake and cargo expression. As shown in the successive images taken from HUH7 cells treated with CKKE12-based particles, by 4 hours, uptake could already be observed with BODIPY, but no mCherry expression was visible until 8 hours post-treatment. Expression continued to increase until it reached a plateau around 24 to 30 hours after treatment. There is a lot more to explore with these lipids and LNP formulations, but I'll wrap up here. We showed in these experiments that Nunchuck can effectively produce both screening and bulk formulations of LNPs. Stunner is a useful tool for initial dynamic light scattering analysis of LNPs and can help optimize the preparation. Cayman has an extensive line of high-quality lipids to customize nucleic acid delivery with LNPs. Pre-labeled lipids such as BODIPY-cholesterol may represent an opportunity to test initial LNP characteristics for screening purposes, but model systems need to be carefully considered. Finally, these data support myriad other data showing that the ionizable lipid used is a critical component affecting targeting of LNPs to tissues and cells of interest. Please take a look at our website for more information on our LNP catalog offerings and services. And at this point, we would be happy to take any questions from the audience.
Thank you, both of you, Julie and Martha. We can now move on to the Q&A. As a reminder, please submit your questions using the Q&A tab to the left of your screen. I see we have lots of great questions already. We'll try to get to as many of them as possible. Let's go ahead and begin. How does Cayman support lipid-based drug delivery?
We can support our customers at all phases of the process, from off-the-shelf and custom lipid synthesis through small-scale formulation and characterization in vitro rather. We also have GMP manufacturing capabilities, so don't hesitate to reach out to us to see how we can help you with your LNP project.
Great. The next question, can you reuse the Nunchuck cartridge?
The Nunchuck is a cartridge that has some potential to be reused. There's nothing about Nunchuck or the Nun that would stop you from using it, but we do rely on our users to use some scientific common sense to use it in a valid way. So we highly recommend that they reuse it within a same day, same chemistry, and be able to run multiple different screening experiments. And if they want to use it beyond that, then we can discuss some ways in that we can optimize that and validate some reuse of the Nun.
I can add on to that actually. For this particular set of experiments, what we did was we generated the minus BODIPY formulations first, and then used the same Nun to generate the plus BODIPY formulations.
Yep, which is a great way of being able to reuse the Nun. Thanks for that, Julie.
All right, our next question. What scale can Cayman operate at for custom lipid and LNP products?
We have a really large offering of catalog lipids, and our expertise allows us to offer custom and novel lipid synthesis as well. We can synthesize anywhere from milligrams to hundreds of grams of high purity R&D grade lipids. We also have GMP capabilities that can go up to kilogram quantities. As for the LNPs themselves, we are using a Nunchuck, so we are bound by Nunchuck's capabilities, which I believe are up to the liter or so capability.
All right, great. For the next question, how much sample does Stunner use up?
Yeah, so for Stunner, our analytical tool, it only takes 2 microliters of sample in order to be able to analyze LNP solutions. And out of Nunchuck, the quantity of LNPs that come out tends to be really concentrated. So in fact, we do a 1 to 10 dilution of the sample and then use 2 microliters of that to analyze it on Stunner. So it's quite a tiny amount of sample. So you can be confident that most of your sample isn't going to go into your analytical method.
Terrific. For the next question, how else can Cayman help with in vitro testing of LNPs?
Well, we have a wide variety of cell lines at our disposal. Really anything that's available from ATCC or really any other vendor. We also have a number of cell lines currently in-house, as well as the expertise to work with either challenging or primary cell types. Additionally, our citation and biospa setup, which I mentioned, allows us to kinetically image up to 8 plates for really any length of time with flexible color formats. And finally, we can test the expression of fluorescent proteins by flow cytometry. And we're also pretty good at customizing assays for really a wide variety of readouts.
All right, great. Our next question, have LNPs made on Nunchuck been validated in animals?
Yes, in fact, they have. Using a collaboration with one of our partners, they have been validated in mice.
All right, thank you. Does the length of mRNA alter LNP parameters?
Yes, it will definitely contribute to the N to P ratio of how you construct the LNP. So basically, what you end up doing is changing the concentration of the mRNA that you're putting in the aqueous phase.
All right, great. The next question. For cell uptake study, did you control under all the same mRNA amount or labeled, there's a word I'm not recognizing, call amount, thank for this one?
I think I understand the question.
Okay.
We for the expression study using mCherry, we did control for the amount of mRNA there. So it was differing concentrations of total particles based on the mRNA concentration that was there. So it sort of makes that CKKE12 and C12-200 even more impressive because there was substantially fewer particles in those cells than because the mRNA is higher per particle.
All right, the next question. I assume the transfection is under the same mRNA amount.
Yeah, that's the same question. Yep.
All right, great. Sorry. What was the dose of LNP used in the mCherry cell expression analysis?
That varied between different, so it was on 100 nanograms per mil was the starting mRNA concentration. We did two dilutions, but I'm only showing one here. But they were consistently kept at 100 nanograms per mil of mRNA.
Great. Do you have experience with functionalized lipids with an affinity-based tissue targeting agents?
We are exploring that right now.
Okay. Can Nunchuck be used outside LNP generation, for instance, to prepare uniform-sized phospholipid vesicles for in vitro enzymatic testing?
Yes. So, as people who work in nanoparticles know, LNPs refer to a specific kind of nanoparticle, but the Nunchuck is a very versatile tool. And as long as you check chemical compatibility, we can make a number of nanoparticles. And what is very unique about Nunchuck is the microfluidics that allows it to have a very reproducible mixing that makes the high quality nanoparticles, but is not limited just to LNPs. Other nanoparticles can be made as well.
Right, terrific. The next question. Can Cayman provide an LNP with payload and empty?
Yes.
Yeah, I would just add to that some lipid formulations are not as stable in the empty form. So while it is possible to create them, sometimes they don't last long, or the nanoparticles are kind of, let's say, wonky sizes and polydispersities because the forces of having the payload inside of the lipid nanoparticle can help hold it together.
I would also add to that, feel free to come to our website. We have a form on there that you can fill out and get in contact with me. And we can have a discussion about what you're looking for and how we can best accomplish your goals.
Okay, wonderful. The next question. Can Stunner be used for analysis of nanoparticles other than LNPs?
Yes, today we focused on Stunner's analysis of LNPs, but Stunner is a highly versatile tool. Again, it only takes 2 microliters of sample, and it has a very wide dynamic range in an analysis using UV vis spectrometry and dynamic light scattering. So it can be used to analyze concentration and quality of RNA, DNA, proteins, different nanoparticles, AAVs. So it is very versatile, and I invite you to check out the brochure that's available as a handout for more information.
Wonderful. That's in the handouts tab at the left side of your screen. You can find that brochure. Does Cayman offer characterization of LNP using EM?
Yes, we do. We can do this in partnership with another a partner that we have that does cryo-EM, and we've actually been doing some of this very recently, and are hoping to be able to share those data very soon.
Okay. Next question: what is the smaller size of LNP that can be made by the Nunchuck?
So that is very chemistry dependent. So there's no specific definitive smallest size that can be made. The sizes that we've seen in some specific chemistries can go down to 50 nanometers or a little bit less than that. But it's dependent on the lipids that are used. For example, some of the lipids, as Julie showed, can have different geometries, and those geometries can be limiting to how big or how small a lipid nanoparticle can be made. And also the payload will affect the size of the LNP.
Okay. The next question. Sorry, I'm scrolling a little bit here. I apologize for the dead air. I am having a little bit of connectivity. Here we go. Sorry about that. Can we add peptides to the lipids formulation to make LNPs with the Nunchuck?
Yeah, yes. There's a couple of different ways to do this. You can include a peptide that is already conjugated to a lipid that is then the particle is then put together in the nunchuck, or you can make your particle with a functionalized lipid and then add your peptide after the particle is made. I think there are various camps of which is preferable, but both are possible.
All right. Well, thank you. We have come to the end of the question and answer period now. Thank you all for attending this Fierce Biotech webinar and for submitting so many great questions for this discussion. I'd like to thank both of our speakers for participating and Unchained Labs for presenting today's webinar. A recorded version of this webinar will be available for your access within 24 hours using the same audience link that was sent to you earlier. Thank you again for joining, and we look forward to seeing you at future events.
Bye.